Today we are going to talk about histology of pancreas, right? Pancreas is basically an elongated gland, right, which is present in C-shape arrangement of the duodenum, right? This part of the pancreas which is larger, that is called the head of the pancreas, and this central part of the pancreas is body of the pancreas, and last this part of the pancreas is tail of the pancreas. So basically pancreas is a gland, elongated gland, right, having a head, body and tail. Is that right? Secondly, pancreas is double gland. It has exocrine portion and it has endocrine portion. Exocrine portion of this gland produces digestive enzymes. Suppose these are the exocrine portion, these are the acini, and these acini, acinar cells release their digestive enzymes into pancreatic ducts, which will be released eventually in, yes, what is it here? In the lumen of duodenum. Is that right? And second portion of the pancreas is endocrine portion. Endocrine portion consists of groups of cells which are called islets of Langerhans, right? These groups of cells, islets of Langerhans, these are different types of endocrine cells which are clumped together, and they are diffusely distributed within the mass of pancreas. And these cells release their secretions like insulin, glucagon and somatostatin. They release their secretions directly into blood stream. So we call that this is endocrine portion of the gland. Now let's look at first of all we'll talk about relationship of pancreatic duct with the bile duct system. So let's suppose, look, here is the liver. From the liver there is right hepatic duct and left hepatic duct, and right and left hepatic duct together make common hepatic duct. And here is gallbladder with the cystic duct, right? Let me explain it more clearly. Here is right hepatic duct and here it is left hepatic duct. Both of them together make common hepatic duct, and here is your gallbladder, and gallbladder is connected right here through the cystic duct, right? And when cystic duct and the common hepatic duct, they come together, right, they make common bile duct. Common bile duct, so it means that common bile duct is extending from the point where cystic duct fuse with the common hepatic duct, and common bile duct come downward and it moves behind the first part of duodenum, and eventually it goes in the back of the pancreas and enters into pancreatic substance. And within the pancreatic substance, this common bile duct, right, fuses with the main secretory duct of the pancreas. So main secretory duct of the pancreas and common bile duct, they fuse together and they make this duct which is called, yes, major pancreatohepatic duct, and which opens in lumen of duodenum through ampulla of Vater, right? And of course you must be knowing that in the ampulla of Vater, at the last part of this duct, there are smooth muscles, and those smooth muscles encircle around this area. We call it sphincter of Oddi, right, which controls the flow of bile and flow of pancreatic juices to the duodenum, as well as this sphincter also prevent the reflux of duodenal content to the pancreas. Am I clear? Another thing which is important to discuss here, that in some patients you will also find accessory pancreatic duct or small pancreatic duct, right, which opens through minor papilla to the duodenum. This actually just reminds you that originally pancreas is derived from two primordial buds which evaginate out of the foregut, right, and later on those two buds fuse together, and bigger pancreatic bud which become the sum of the head, what is this? Body and tail, that is drained by the major pancreatic duct, and smaller portion of the head, right, which is derived from the smaller evagination from the foregut, that may be drained in some people independently through the accessory pancreatic duct. Do you have any question here? Now let's go into detail. What I'm showing in this diagram, that here this is the ductal system, right? Let's suppose this is a major duct, right? This is major pancreatic duct. Now this duct, actually in the major duct, these ducts are opening, and these ducts are basically, what are these ducts which are directly opening into it? These are interlobular ducts. Interlobar duct, and then they are extending and they are draining basically interlobular duct, right? So what are the ducts? Let's suppose if this is the major duct, this is the major pancreatic duct, right? Now here what is draining? Lobar duct, and then what are these which are draining into interlobar duct? These are interlobular duct. So these are inside the lobules, interlobular duct, and interlobular ducts actually drain intercalated ducts. These are intercalated duct, and this part of the intercalated duct which is extending into the acinus. This is very important to understand. These are the acinar cells, acinus of the gland, which is usually, of course, these are the cells, pyramidal cells, which are simple cuboidal epithelium. So secretory portion of this gland, which is called acinus, right? This is maybe acinus or tubuloacinar gland, right? And these cells are pyramidal cells which are narrow on the luminal side and wide on the basal side, and basal side is towards the blood side, and luminal side is releasing the digestive enzymes where into the lumen, right? Now this acinus is like a balloon, and this intercalated duct is like a straw, you put into a balloon from where you can drink. Is that right? In the same way, in other glands, actually these ducts should start here, but the very unusual and unique feature in pancreatic exocrine portion is that these intercalated duct cells extend inside the lumen of acinus, right? And these cells of the duct which are extending into the lumen of acinus, these are called centriacinar cells. What are these cells? These are centriacinar cells, right? But they are part of intercalated duct. What is this duct? This is intercalated duct. So now let's reverse in direction. That exocrine portion of the gland consists of special acini and draining duct system. These acini are basically made of pyramidal cells, is that right? And they secrete the digestive enzymes into the lumen. Then within the lumen, right, there is formation of, what is this? Intercalated duct. And those cells of intercalated duct which are present protruding in the lumen of acinus, they are called centriacinar cells. And these ducts, right, intercalated ducts especially, they're very important to remember. You know why? Because they secrete bicarbonate ions, right? Intercalated ducts are responsible to release what? Bicarbonate ions. If intercalated cells of the intercalated ducts are stimulated either by the acetylcholine or they are stimulated by the secretin. You must be knowing that secretin is produced by the S cells of the intestine, and secretin through the blood will reach to the pancreas and act on the pancreatic ductal cell, especially which cell? Which duct? Intercalated duct. And intercalated duct cells, when they are stimulated, they produce profuse amount of watery alkaline secretion, because these cells, ductal cells, secrete a lot of bicarbonate. But these acinar cells do not secrete the bicarbonate. These cells are usually stimulated by acetylcholine or cholecystokinin and acetylcholine. So we can say acetylcholine from cholinergic nerve endings for parasympathetic nervous system stimulate the acini, as well as acetylcholine can stimulate the ductal. So acetylcholine can stimulate the acinar cells and increase the release of enzymes, as well as acetylcholine can stimulate the ductal cell and increase the release of bicarbonate. But cholecystokinin specifically stimulate, what is this? Acinar cells, and it produces enzyme rich secretion, but cholecystokinin does not significantly stimulate the intercalated ductal cells. So cholecystokinin does not increase the secretion of bicarbonate. So what we can say, cholecystokinin is main stimulant of acinus of the pancreas, exocrine pancreas, and they release enzyme rich secretion. And what is this here? Secretin coming from the which cells? Sorry, secretin is coming from the S cell, we should correct it, and cholecystokinin is coming from the I cell of the intestine. Again, cholecystokinin from I cells secretin coming from the S cells, but both of them are coming from GIT. Cholecystokinin does many functions. One of the function is the cholecystokinin stimulate the acini of pancreatic, exocrine portion of the pancreatic gland and increase enzyme-rich secretion. And secretin specially stimulate the intercalated ductal cells and increase bicarbonate-rich secretion. But if there is combined action of cholecystokinin and what is this? Secretin, then exocrine portion of the pancreatic gland produces copious amount of secretions, large amount of secretion, which are very rich in enzyme and of course very rich in bicarb. Is that right? These bicarb are important because these bicarb which are produced by these ductal cells, when they come over here, they help to neutralize the acidic chyme which is coming from the stomach and adjust the pH of duodenal content appropriately so that which enzyme can work? Pancreatic enzyme can work, because gastric enzyme work in acidic environment better and pancreatic enzyme work in alkaline environment better. That is why. And secondly, in this diagram you can see that all these ducts are moving over there, and what are these? These green are the acini, right, which are secreting. Plus, what are these red things? These are islets of Langerhans. I will talk about them later. Now we come to in detail of these acinar cell structure. These acinar cells are very simple cell. These are having all the characteristics of those cells which synthesize a lot of proteins and secrete them. Because this is nucleus is pushed a little bit on the basal side, and because these are proteins synthesizing and secreting cells, so they must have a very prominent machinery to synthesize the peptide chains. That is rough endoplasmic reticulum. So these cells are very rich in rough endoplasmic reticulum and even of course endoplasmic reticulum loaded with the ribosome, and it may have some free ribosomes also. And due to this rough endoplasmic reticulum loaded with ribosome and free ribosomes which are very abundantly present, this cell stains in a basophilic color. So basophilia seen in these cells is due to rough endoplasmic reticulum and free ribosomes. Am I clear? Plus, on the apical side, you find a very prominent Golgi apparatus. You know, this is Golgi apparatus is very visible in every cell, right? This is basophilia, this blue color, and Golgi apparatus. And this Golgi apparatus is concerned with concentration and packaging of, and post-translation packaging of, what is this? Peptides, which are of course enzymes. And eventually, once these enzymes which are synthesized here, actually this initially synthesizes proenzymes, right? And these proenzymes are processed within the Golgi apparatus, and then these are ready to be secreted, and for secretory purpose they are packed into special membrane bound vesicles which are called secretory vesicles, or which are also called zymogen vesicles. And these vesicles are very rich in enzymes. Is that right? So whenever stimulus come, then of course these vesicles will be secreted on the luminal side, and enzyme will move through the ductal system to the small intestine or duodenum. Any question you have here? Then of course you can see mitochondria are scattered throughout, right? Or that cell is clear to you, as well as this acinous structure, right, and ductal structure.
Enzymes are mucus type or serous type?
Yeah, these are all actually, this is a serous gland, right? This glandular structure, this is not a mucus gland, it's a basically serous gland, and its structure is very similar with the parotid gland. The easy way to differentiate on the slide, that is it a parotid gland or exocrine pancreatic gland, major difference which you can see in parotid gland, these ducts do not extend into the lumen of the acinus. So these centroacinar cells are positive or present in exocrine portion of, what is this? Pancreas, but they are absent in parotid gland. Do you have any question? So this is basically a serous gland, right? Now we come to this portion of this gland, that this portion of the gland I have just highlighted or enlarged one, what is this? Islet of Langerhans, but there are a lot of islet of Langerhans diffusely present within the parenchyma of the gland, right? This islet of Langerhans, they are having cells which are endocrine cells, and by special technique, if you stain them, central cells are stained orange brown color, brownish orange color. These are called beta cell or B cells, and they are responsible to secrete the insulin into blood, which reduces the level of glucose in the blood. and then on the outer periphery we can see red stained cells. These are called alpha cells or A cells, right? And alpha cells are responsible to produce glucagon. Glucagon is produced when blood glucose levels are down, and it increases the blood glucose level, and it also increases free fatty acid concentration in the blood, and it is ketogenic, right? But remember, insulin, which is produced by the beta cells, the B cells, it is produced when blood glucose levels are high, and it actually reduces blood glucose level by increasing the uptake of glucose into peripheral tissues, right? And then these blue cells are basically delta cells, and these delta cells produce somatostatin, and this somatostatin is responsible to inhibit the oversecretion of insulin by beta cell or oversecretion of glucagon by alpha cells, right? So I will not go into detail of insulin and glucagon because you will study that in very big detail in physiology and biochemistry, right? Let's go back about little bit clinical points related. Oh yeah, let's talk about the enzymes which are secreted. The enzymes which are released over here, there are some enzymes which are for the proteins. Is that right? Enzymes which are proteolytic enzymes coming from the pancreatic system, exocrine gland. Number one, there are exopeptidases. Exopeptidases. And there are endopeptidases. Okay, let me write endo first. Endopeptidases and exopeptidases. Endopeptidases are those enzymes which can break the peptide chain from within, but exopeptidases are those which can remove the amino acid from the ends of the peptide chain. Now, classical example of endopeptidases which are secreted by the pancreatic acini, that is trypsinogen, which is inactive form, and chymotrypsinogen, trypsinogen. Now, trypsinogen and chymotrypsinogen are your endopeptidases, and exopeptidases are some work on carboxyl end of the peptide, other work on the amino end of the peptide chain. Those which work on the carboxyl end of the peptidase, we call them pro-carboxypeptidases, pro-carboxypeptidase, and those which work on the amino end, they are called pro-amino, yes, peptidases, right? Then we come to those enzymes which are related with help in digestion of fats and lipids, right? Lipids. In that case, classically we have lipases, pancreatic lipases, we have pancreatic cholesterol esterases, steryl esterases, phospholipases, right? And then we have enzyme for the carbohydrates, and that group of enzyme is basically pancreatic amylases. So how many we have? Trypsinogen, chymotrypsinogen, pro-carboxypeptidases and pro-aminopeptidases, all of them are pro-enzymes, and they are concerned with digestion of proteins, right? These four enzymes, fifth and sixth, lipases and cholesterol esterases, right? They are mainly concerned with digestion of lipids, right? So then of course lipases include phospholipases also, and then amylases for the carbohydrate. How many enzymes? 4, 5, 6, 7. Then there are enzymes also for digestion of nucleic acids, right? So those enzymes which digest which nucleic acid DNA, they are called deoxyribonucleases. Ribonucleases or DNases. And then there are those pancreatic enzymes which break down the RNA, and we call them ribonucleases. Ribonucleases. Am I clear? Now, this is very important function of the pancreas, that it secretes a lot of digestive enzyme. Here, there are special enzymes which are expressed on the surface of the duodenal cell, the gastrointestinal cell. These enzymes are called enterokinases. Enterokinases. Now, this is very important to understand, that as all these enzymes are coming as inactive thing, because if they are activated within the substance of pancreas, they will start autodigestion. So this inactive enzyme, when they come over here, let's suppose trypsinogen, trypsinogen. Now enterokinases will work on trypsinogen and convert it into trypsin. So trypsin is the active form. Now trypsin, not very important function of activated enzyme trypsin is that then trypsin will attack other enzyme coming from the pancreas and activate them. So what is the first step? Enterokinases activate the trypsinogen or trypsinogen into trypsin, and then trypsin will act on other pancreatic proenzymes and activate them. Am I clear? Any problem up to this? Another point which I would like to highlight here, if pancreas is damaged, right, injury occur, there may be acute pancreatitis or chronic. Acute pancreatitis is severe injury to the pancreas where pancreatic enzymes are released into pancreatic substance and activated over there, and autodigestion of pancreas start. That produces severe abdominal pain, severe central or epigastric abdominal pain which radiate backward, right? And with this, it presents like an acute abdomen, but actually it's a medical condition. And in this case, pancreatic enzymes will spill over into circulation and also appear into urine. So one of the way to diagnose is that in the blood of this person who has acute pancreatitis, there will be high level of amylases, pancreatic amylases will be there, and pancreatic lipases will be there. In the same way in the urine also pancreatic amylases and lipases will be there. And in pleural fluid of these patients who have acute pancreatitis, there are also high levels of amylases which are diagnostic of acute pancreatitis, right? And in acute pancreatitis, as these lipases, pancreatic lipases are released from here and act on the fat, peritoneal fat, and they damage the, partially digest the peritoneal fat, and free fatty acids which are released, they bind the calcium, and these fats, partially digested fat with exposing negative charges of fatty acids, right, there the calcium bind and calcification points will be seen where in abdominal peritoneal cavity where the damage to the fat has been done by the pancreatic lipases. Am I clear? Then some people unfortunately develop remitting, relapsing chronic pancreatitis. And over the year, if chronic pancreatitis becomes more and more progressive, most of the exocrine function of the pancreas is lost, what will happen? There will be severe deficiency of pancreatic enzyme. And the most important deficiency will be lipases. And person will develop maldigestion and malabsorption problem, because if pancreatic enzymes are not there, you are unable to digest, especially lipids well, because proteins may be digested by other enzymes also, carbohydrates may be digested by other enzymes also, but severe type of fat malabsorption occur, and that produces fatty stool which is called steatorrhea. These stools are basically fatty stools which are produced due to pancreatic insufficiency. They are, what is this? The bulky stools, foul smelling, float in the pan, difficult to wash away, right? All those features. And of course, if someone has chronic pancreatitis from early childhood, he will also have stunting of growth. One of the cause of chronic pancreatitis may be the people who have a lot of alcoholic, they take a lot of alcohol, plus people who are having the gallbladder stones. Is that right? Do you have any question here? And anything? Nothing left there? So these are a few words about the histology of pancreas and detail of islets of Langerhans you will study when you will be studying the endocrinology in biochemistry and physiology. Class dismissed.